Thank you, Fatia, for that presentation. So now I will continue with my part, which is on the mechanism of cell-mediated immunity. So in the mechanism of cell-mediated immunity, we have four steps actually. The first step is the recognition of foreign antigens. The T lymphocyte will respond to a distinct antigenic determinant. An antigen selects a specific pre-existing clone and activates it. And then step 2 is the activation of T lymphocyte. The macrophage will capture, engulf, and digest the antigen and present it on its surface. The activated T helper cells will secrete cytokines and activate cytotoxic T cells, B cells, macrophage, and natural killer cells. The T helper cell will proliferate into either T helper 1 or T helper 2. The third step is the antigen will be eliminated by the effector mechanism. The activated Tc will proliferate and differentiate into a mature Tc. The MHC1 of infected cell surface bind with the CD8 receptor of Tc. The secretes perforins, granzyme protein, and lymphatic toxins. The activated Tc will proliferate, forming a clone of cells, effector cells, and eliminate other infector cells. And the last step is it will return to homeostasis. The immune system will return to its basal resting state. It will balance itself. So, what is the relationship between cell-mediated and humoral immunity? So, we have T-dependent antigens and T-independent antigens. For the T-dependent antigens, the antibody production requires assistance for T helper cells, while the T-independent antigen does not require assistance from the T cells. For the T-dependent antigens, a macrophage cell ingests antigens and then presents it to the Ty cells. The T helper cells stimulate the B cells specific for the antigen to become plasma cells. And then antigen are mainly proteins on viruses, bacteria, foreign red blood cells, and heptan carrier molecules. Well, for the T-independent antigens, the antigens are mainly polysaccharides or lipopolysaccharides with repeating subunit, usually bacteria capsules. It have a weaker immune response than for T-dependent antigens. Sorry. So the major difference between cell-mediated immunity and humoral immunity we can see on the slides. For the cell-mediated immunity, it is mediated by T cells, no formation of antibodies, secret lymphokines, receptor, and used to detect antigens. And the rest you can read on the slides. For humoral immunity, it is mediated by B cells, formation of antibodies, no secretion of lymphokines, and antibodies are used to detect antigens. And one of the more obvious differences in the cell-mediated immunity, it is slow delayed response, while in humoral immunity, it is a more quick response. So for the importance of cell-mediated immunity is we have four, which is to detect and destroy intracellular pathogen, destruction of tumor cells, delayed type hypersensitivity reaction, and vital for graft rejection. I think that is all from us, from the group, from our group, and thank you for taking your time for listening our presentation. That's all for me.